This video is going to be about inflammation and fever. So inflammation we'll start with. This is a little simpler than fever. So inflammation is described as local because it's not a full body event usually, right? Usually you have inflammation of a specific cut or specific region of your body. So it's local. The symptoms are, as you know, redness, pain, heat, swelling. We usually try to dampen the effects of inflammation, but it actually is there for a reason. So it helps you fight disease. The function of inflammation is to destroy injurious agents, to confine the infection, and to speed up tissue repair. So it's actually not ultimately good for your body to dampen inflammation unless your inflammation has run wild and is no longer no longer benefiting you, no longer serving you well. But under most situations, we want inflammation. It actually helps us prevent infection and speed up tissue repair. So this is how inflammation is initiated. You have microbial PAMPs, which stimulate TLRs on macrophages. So remember, PAMP is pathogen-associated molecular patterns. Those are those generalized molecules that are common to many different kinds of pathogens, because we're talking about the innate or the non-specific immune response right now. This is your immediate and non-specific immune response. So you have PAMPs, pathogen-associated molecular patterns, stimulate TLRs on macrophages at the site of a wound or at the site of an infection. So TLRs are the toll-like receptors that read those generalized molecular patterns. Those macrophages will then become activated and they're going to release cytokines. So for example, they will release TNF-alpha, tumor necrosis factor alpha. And that's one of the three classes of cytokines I wanted you to know from a previous lecture video. TNF-alpha, tumor necrosis factor alpha, causes the liver to release acute phase proteins. So you have the macrophages at the site of infection or at the site of a wound, activated by microbes and releasing TNF-alpha. TNF-alpha travels through the bloodstream to the liver, and it causes the liver to release acute phase proteins. And acute phase proteins do a whole bunch of things. They activate other acute phase proteins. Acute phase proteins include fibrinogen, which we've discussed before. So remember, fibrinogen is floating around in the blood, and when they become fibrin, they create these long networks and nets that help stop you from bleeding out, that create blood clots. So fibrinogen is a protein that helps with blood clotting. So acute phase proteins include this blood clotting protein, fibrinogen, and they also cause vasodilation. So vasodilation is a widening of the blood vessels. Okay, so I'm not going to test you on acute phase proteins. You can just skip that step and just know that TNF-alpha is responsible for those. Okay, because we can just, TNF-alpha, this cytokine, it's an important cytokine that is involved in inflammation, and the results of it are vasodilation, or the widening of the blood vessels, blood clotting with fibrinogen, and activating other acute phase proteins. So there's other acute phase proteins that play different roles that we won't cover. Also, by activating a whole bunch of them, you get this cascade that makes the signal louder and louder and louder, so that you don't just get a little bit of inflammation, but you get enough inflammation. A small signal can create a lot of inflammation in order to really help fight infection and heal. So let's look at the stages. So just to recap, before we go into the stages of inflammation, we have pathogen-associated molecular patterns on the microbe stimulating toll-like receptors on macrophages, which is a kind of immune cell of the innate immune system. The macrophages are now activated. They're releasing cytokines like TNF-alpha. TNF-alpha is going to travel to the liver, releasing acute phase proteins like those that cause blood clotting and those that cause vasodilation, among others. Stages of inflammation. So I know that you'll look this up on Google and I'll get a whole bunch of different answers in the homework about the stages of inflammation. I would like you to know these three. I'm really trying to distill this down to the most critical concepts. And of course, for these complicated biological processes, you can divide up the processes in a million different ways. I would like to keep our understanding consistent, so please learn it this way, these three stages of inflammation. So first stage I want you to know is vasodilation. So vasodilation includes increased permeability of the blood vessel walls. So Vasodilation means you're making the blood vessels wider. So that's why the area gets swollen. So you get a cut and it gets swollen and red. That's because of vasodilation. So the blood vessels have dilated, meaning they've become wider. That means there's more volume in the blood vessels to bring more blood. So the area gets warm because the blood is warm coming from your core. Your core is very warm and brings the blood brings that warmth with it as it travels to the site of inflammation, the site of infection, the site of wound. So it gets warm to the touch, it gets red because of that extra blood, and it gets swollen because of that extra blood, which involves a lot of liquid. So vasodilation brings more blood to the area and is responsible for a lot of the symptoms you have with inflammation. And then it also includes increased permeability of blood vessels. So we talked in the video about the different kinds of cells of the innate immune system. We talked about neutrophils, which are phagocytes that can leave the blood vessels. Well, if the blood vessels if the cells that make the blood vessels are really tightly packed, it's harder for them to leave the blood vessels to go to infected tissue or wounded tissue. So increasing the permeability of the blood vessels makes it easier for those neutrophils to leave the blood vessel walls and go directly to infected tissue to help clean up any infection. Okay? So vasodilation and increased permeability go together. Then you have phagocyte migration and phagocytosis. So we have all different kinds of phagocytes. We have the neutrophils, we have antigen-presenting cells. So all of these phagocytes move through chemotaxis, so they're attracted to the site of the wound and to the site of any kind of infection that might be there because of chemical signals. So wounded cells will release chemical signals that they are wounded that will attract phagocytes. Also, microbes themselves release chemicals that phagocytes are attracted to. So that's chemotaxis, it's the movement in response to a chemical gradient, right? So the phagocytes migrate attracted through the process of chemotaxis to the site of infection, and then they phagocytize any debris. So they'll phagocytize anything that's foreign. They'll phagocytize, they might not just phagocytize pathogens, they may phagocytize debris as well. So when you get a splinter, that wood gets degraded over time even if you don't pull it out. It'll get red and inflamed and it'll hurt, but almost always it will just disintegrate in your body. There's a chance that it could introduce infection, but it almost always will just disintegrate, and that has to do in part with phagocytes breaking it down. And then the third stage is tissue repair. So tissue repair cannot be completed until all the injurious agents are removed. So if you have a splinter that's in the way, your tissue is not going to completely heal until that's removed, right? Tissue repair starts right away, but it cannot be completed until until step 2 is finished, until the phagocytes finish cleaning up all the dirt, all the debris, all the pathogens that may have entered through the wound. So the reason for vasodilation is I told you that it brings a lot of blood to the site of infection. The reason that's good, there's a few reasons that you want to bring more blood to the site of an infection or to the site of a wound. One reason is you want to bring more oxygen because you're going to have a lot of white blood cells and they're going to be working really hard. They're going to be metabolizing a lot, and they use aerobic respiration. So just like when you're working really hard, you need to breathe more, breathe harder, breathe more. Phagocytes also, when they're doing their job, when they're working, when they're really active, they're going to be burning through that oxygen faster. So you need to bring more oxygen to the site of the injury or to the site of infection so that the white blood cells can do their jobs more effectively. And then you also, and also you're bringing nutrients, of course, and everything like that when you're bringing more blood to a site of infection. But also you just want to bring the blood cells themselves. And you also want to bring platelets, and you want to bring those fibrinogen. You want to bring those blood clotting proteins. So and cell fragments like the platelets. So when you bring blood to an area, when you bring more blood to an area, you're making everything work faster. You're bringing more white blood cells, you're bringing more blood clotting agents, and you're bringing more oxygen and nutrients to facilitate healing and to facilitate tissue repair. So you're basically bringing all of the builders, the healers, the fighters, the building materials, all of that stuff is getting to the site faster when you dilate the blood vessels to bring more blood to an area. And fever. So fever and inflammation they're similar in some ways, but they have some important differences. So fever is systemic, meaning it's your whole body, right? When you have inflammation, it's one specific area of your body that's infected or that's wounded. A fever is your whole body. So the way you get a flea fever is in your brain you have a thermostat, and the piece of your brain that holds your thermostat is the hypothalamus. So just like on your wall, the thermostat on your wall, what it does is it senses temperature, and you have a setting for the temperature that you want your house to be. And so the thermostat on your wall senses the temperature of the room, and it compares it to the thermostat setting. So if you said, okay, I want my room to be 72 degrees Fahrenheit. So you put 72 degrees Fahrenheit into your thermostat. The thermostat senses when the temperature rises above or below. And if you have your thermostat, let's just say the thermostat in your body is linked up both to cooling and to heating mechanisms, and the thermostat in your house maybe as well. So if the temperature rises above 72 degrees Fahrenheit, the thermostat will switch on a cooling device like your swamp cooler or your air conditioner. And if the temperature dips below 72 degrees, your thermostat in your house will kick on your heater. Now, the thermostat in your body does the exact same thing. So in your hypothalamus, in your brain, is a thermostat, and it's set to somewhere around 98 degrees Fahrenheit, right? 98 point something degrees Fahrenheit. So that thermostat setting, it's also constantly sensing your body temperature. So if your body temperature gets up too high, your body will turn on the cooling mechanisms, which include mainly sweating. So the heat from your body leaves in your sweat. It's evaporative cooling, just like a swamp cooler. And then if your body temperature dips below that 98 degrees Fahrenheit, you'll initiate your heater will turn on, and that includes things that increase your metabolism. So your metabolism will ramp up, and so, for example, you'll start shivering. Shivering is a lot of fast muscle contractions. That gets you metabolizing faster to warm your body up, right? Because the faster you're metabolizing, the more you're burning through calories. It's like burning through a log, right? You're burning calories. That's literally creating heat, just like burning a log is burning calories and creating heat. So the thermostat in your body is in your hypothalamus, and it's linked up to both cooling and heating mechanisms. And your behavior is also cooling and heating mechanisms. So you might shed clothing when you're feeling too hot to bring your temperature down. You might add clothing or blankets when you're feeling too cold to heat up. Now, when you get a fever when you're sick, what's happening is your body resets the thermostat setting. So it's like instead of being at 98 degrees Fahrenheit, your body, in response to infection, will reset that thermostat setting to maybe 102 degrees Fahrenheit. And then when you take your temperature, you're 102 instead of 98. Okay. So the symptoms of fever, of course, are hot body temperature, but also clammy skin. And we'll talk about why in the next couple slides. And you also get alternating shivering and sweating. And the shivering and sweating, that's your cooling and heating mechanisms to help you maintain that higher thermostat setting. And also as the thermostat setting changes, because when you're sick, the thermostat setting keeps fluctuating, and it's in response to microbial signals in your body or what your immune cells are telling your hypothalamus to do. So your white blood cells and also chemical signals from the bacteria or virus in your body is constantly changing this thermostat. So maybe it's 102 and then the thermostat setting changes again to 100, then you need to sweat to cool down, but then it might go back up to 102 again, so then you need to shiver in order to heat up again. Right? So the function of fever is to improve the function of immune system proteins, to increase the speed of tissue repair, and to reduce the activity of temperature-sensitive microbes. And I'm going to go through each of these. So, improve immune protein function. Let's talk about that first. So there are many proteins in the immune system that function better at temperatures that are just slightly above normal body temperature, slightly above 98 degrees Fahrenheit. So for example, we talked about interferons before exam 3. So this is the slide from Microbial Mechanisms of Pathogenicity PowerPoint on interferons. So interferons inhibit RNA and protein synthesis inside the cell and inside neighboring cells. So a microbe can't use the cells to produce its own proteins and enzymes. So this is specifically in response to viral infection, right? Because bacteria are more self-sufficient because they're actually live bacteria, have their own transcription translation equipment. A virus does not. The whole reason a virus infects the cell is to hijack transcription translation equipment in the cell. So a virus has to use the cell's transcription translation equipment in order to make new viruses. So if the cell shuts down its own transcription translation, the virus cannot replicate. So in response to viral infection, our cells produce interferons, which shut down transcription translation, and they release those interferons to neighboring cells to shut down transcription translation of the neighboring cells, hopefully before the virus gets there. Now this kills the host cell because the cell also needs transcription translation to survive. Now these interferons actually work better at temperatures slightly above body temperature. They work better with fever. Okay? And there's lots of proteins of the immune system that function better at slightly higher temperatures. This harks back to optimal temperatures that we talked about early in the term, because these are all proteins, and proteins have that complex folding pattern, and that folding pattern can be disrupted by temperature. So if it gets too hot, they'll denature, but if it's too cold for that particular protein or enzyme, it will be too rigid. It won't work so well. And so depending on the structure of the protein, it will have different proteins have different optimal temperatures. The proteins of your immune system work better at higher temperatures than at body temperature. Many of them do anyway. All right, so increase the speed of tissue repair. So why would increased temperature increase the speed of tissue repair or improve function of proteins such as interferons? So remember that, that tissue repair is being performed by enzymes as well, which are proteins. So enzymes are the catalysts for every chemical reaction, including the chemical reactions involved in building new tissue. If you're building tissue, you're building big molecules out of smaller ones. Those are chemical reactions catalyzed by enzymes, which are almost 100% of the time they are proteins. So let's go back to this slide that I was referencing. So again, here's enzyme activity, how fast a particular enzyme is working, and here's temperature, going from 10 to 50. As the temperature rises, the enzyme is working faster, faster, faster. And this is true for all enzymes. Enzymes at first will start working faster, faster, faster as you increase the temperature. And that has to do with them encountering their substrates and because we talked a little bit about how an enzyme, once it finds its substrate, it changes shape slightly in order to change the substrate into product, and then it bounces back to its original shape once the product is released. That malleability, that ability of the enzyme to move a little bit, that's always going to work better at higher temperatures. Basically, the warmer something is, the more energy is in the system, and that facilitates movement. So the warmer it is, the faster enzymes and substrates are floating around, the more they collide with each other, so the more often an enzyme encounters its substrate to turn it into a product. And also the enzyme itself is going to be more flexible and move more easily. So you warm it up, you get faster and faster enzyme activity until you reach a particular point at this is its optimal temperature. After the optimal temperature is reached, its activity starts to decline as temperature increases. And that's because the protein is denatured. There's so much energy in the system that it's disrupted the chemical bonds that hold the protein's shape together. Right? So this pattern is true for every enzyme. What the optimal temperature is different for every enzyme. So for some enzymes, the optimal temperature might be down here. For some other enzymes, it might be up here. And it turns out that every enzyme actually tends to have, this is for a vat of enzymes, but if you looked at an individual enzyme, every enzyme's temperature range tends to be fairly narrow. And so for this reason, pathogens often have a very narrow temperature range in which they can function. So this explains why a very high fever can kill you. If your fever goes above the range of temperatures that your enzymes in your body can tolerate, they will denature, right? But you can also denature the enzymes of the pathogens inside you with fever. So fever is sort of like a game of chicken. Whose enzymes are going to denature first, yours or the pathogens? Okay. So when we're talking about speed of tissue repair, speed at which enzymes are, white blood cells are working, speed at which proteins of the immune system are working. A fever tries to get us to the optimal, we try to bump up our temperature a little bit to reach the optimal temperature for our immune system proteins for repair and for fighting disease, but we also are trying to bump ourselves over the optimal temperature for the enzymes of the particular pathogen so that the pathogens are weakened while our immune system is strengthened. And I'm going to remind you what we talked about syphilis earlier in the term. So syphilis is an obligate biotroph. An obligate biotroph is, so obligate means, like it has to be, it's necessarily a biotroph. And a biotroph is something that lives as a pathogen or a microbe that lives inside its host. So an obligate biotroph has to live inside of its host to survive. It does not live outside of its host. It needs direct contact in order to transmit because it cannot live outside in the natural environment. And syphilis is the microbe, the bacteria that causes syphilis is very, very temperature sensitive. But its optimal temperature is just a little bit below our body temperature. So actually its preferred temperature is slightly, its temperature range includes body temperature, but its optimal temperature is slightly lower than body temperature. And so to fight syphilis, before we had antibiotics, we used to use malaria therapy because fever can kill this bacteria, Treponema Politum. So its optimum temperature is a little bit below body temperature. Optimal temperature is 91 to 95 degrees Fahrenheit. Normal human body temperature is 98.6 degrees Fahrenheit. So let's look at the next slide is going to be in Celsius. So its optimal temperature is 33 to 35 Celsius. Our normal body temperature is 37 degrees Celsius. So if you bump this body temperature with a fever, it's going to weaken or maybe even kill Treponema Politum. It's definitely going to give your immune system an edge over it. So we would inject the parasite that infects red blood cells. We would inject malaria into people with syphilis. And then you can see malaria is described by these cyclical temperature spikes. And these temperature spikes up to 41.5 degrees Celsius when the optimal is just 35 for syphilis. So now after we developed penicillin, penicillin worked to kill syphilis as well, and then you didn't have to have malaria. So that's obviously preferential. But before penicillin, this was the general treatment for syphilis. And then you have to deal with the malaria afterward, but at the time that was easier than dealing with syphilis. So these temperature spikes are high enough to get rid of syphilis. So this is to say, if I go back a few slides here, so that covers reducing the activity of temperature-sensitive microbes as well. So all of these points on here is, they're all related. They're all related to this temperature graph. So this temperature graph is shifted slightly to the right. The optimal temperature is slightly to the right for your immune system proteins and slightly to the left for most pathogens. And this means that fever is beneficial. It's really uncomfortable. We really don't like it. We like to take fever reducers, but the fact remains that it helps you fight disease. And many studies have shown that taking fever reducers actually prolongs the course of a disease. So when you take a fever reducer or you give one to your child, they're likely going to actually be sick longer than they would have been if you just let them suffer through the fever. And now I have to make a point here. Remember, I said that a high fever can be dangerous. And so that's the balance you want to strike. Most people are just out there thinking fever is just 100% bad. It's caused by the microbe. It's not. It's caused by your body, and it's a healthy reaction to infection. So what you're really trying to balance is having enough of a fever to help you fight disease, but keeping it under what's dangerous. And for children, that's especially important because they can get febrile seizures, and so a lower temperature is dangerous to them than is to us. So as adults, we can tolerate having a higher fever than children can tolerate. Okay, so how fever is initiated. So your normal body temperature is about 37 degrees Fahrenheit. So that's your normal hypothalamus thermostat setting. Then there's a lot of ways that fever can be initiated, but let's just go through basically fever, this point here, I'm writing phagocytosis of a gram-negative bacteria, endotoxin, the lipopolysaccharide from their outer membrane is released. That's one way, but there's actually lots of chemical signals that can start this process. It's not only endotoxin, it's not only the outer membrane of gram-negative bacteria that can initiate fever. Viruses can initiate fever, cytokines, basically anything that causes a certain kind of cytokine release, like TNF-alpha, can initiate fever. Okay, so this is one way that fever could start. So let's just say you have an infection from you got your normal temperature setting in your hypothalamus, and then you get infected by a gram-negative bacteria. So it's got that outer membrane made of lipopolysaccharide. We call that endotoxin. So your phagocytes encounter this pathogen, phagocytize it, spew out the remains, and that includes that endotoxin, which is endotoxin or lipopolysaccharide. That's one pathogen-associated molecular pattern, right? That's one type of PAMP. It's a generalized molecule common to all gram-negative bacteria. So that PAMP is going to stimulate a cytokine release from some of your immune cells, including TNF-alpha, tumor necrosis factor alpha. So we have that same cytokine again that we saw with inflammation. So TNF-alpha is then going to travel, if you have enough of it, it's going to travel to the brain, and the amount of TNF-alpha is going to reset, so TNF-alpha is going to reset the thermostat in your hypothalamus to a higher setting. And the amount of TNF-alpha is going to determine how high that thermostat setting goes. So maybe now you've got all this TNF-alpha from all this endotoxin that's being released. That's going to reset the thermostat, let's just say to 100 degrees Fahrenheit, 102 degrees Fahrenheit or something like that, or 40-ish degrees Celsius. So then you get vasoconstriction, increased metabolism, and shivering. This is all going to heat up your core. It's going to raise your body temperature, but it's also going to make you feel chilled. So let's break that down a little bit. Vasoconstriction means the opposite of vasodilation. So instead of widening the blood vessels, we are narrowing them, but only in the extremities. So you get vasoconstriction in the extremities. That's to keep all the blood at your core. So you don't have as much, so when the blood is at your skin and at your extremities, it's releasing heat to the external environment. If you keep all that blood in your core, it's going to retain your heat better. So that's why your skin feels clammy even when your body temperature is rising, because the blood is being pulled away from your skin to keep it in your center to keep you warm. So you get clammy skin but a high internal temperature. Increased metabolism, you start burning through those calories and you start shivering. So remember I said the purpose of shivering, you're clenching your muscles really tight, and that's causing them to burn through oxygen, burn through your calories and heat up. So you're burning these calories faster, you're heating up. So you're actually heating up when you feel chilled. The reason you feel chilled is because you started at 37 degrees Fahrenheit and we've reset the thermostat to 41 degrees, or excuse me, your thermostat was 37 degrees Celsius. We've reset the thermostat to 41 degrees Celsius. So now your natural body temperature is under the thermostat setting. So that's going to tell your brain that you're cold. So you're going to feel cold and do all the things you would do when you're cold to turn up the heat. Just like the thermostat in your house. When the room temperature, if the thermostat in your house is 72 degrees Fahrenheit, the room temperature dips below that, the thermostat's going to kick on the heater. It's going to say, oh, the room's too cold. Let's pick up the heater. That's what your body's doing. So even though your body temperature is in the process of rising, it's going from 37 to 38, 39, until it reaches the new thermostat setting of 41 degrees Celsius, you're going to feel cold because your brain only senses that your body temperature is lower, colder than the thermostat setting. So you feel cold and your body ramps up all of its heating mechanisms until it reaches the new thermostat setting. So that's what a fever is. Then the cytokines eventually disappear, right? So you fight the disease, the bacteria is gone, the endotoxin's gone, TNF-alpha is gone. Now the signal to ramp up the thermostat is gone. So the cytokines disappear and your thermostat lowers back to normal, the normal 37 degrees Fahrenheit or Celsius. So then you have to cool down, okay? And your cooling mechanisms are sweating. That's what you always do when you're too hot. So you're going to feel hot. You're going to want to shed clothes. You're going to start sweating. You're going to feel like you're burning up. That's when you're actually starting to cool down because the thermostat setting has lowered back to normal. Now you're at 41 degrees Celsius and you're going down to 40 to 39. You're going to feel too hot until you go back to 37 degrees Celsius because your body temperature is above the new thermostat setting of 37 degrees Celsius. Okay, so when your body temperature is coming down, that's when you feel hot because your body says, turn on the cooling mechanisms, we're hotter than the thermostat setting. When your body feels cold, that's when your fever is actually rising. So it's a little bit counterintuitive. How you feel when you're sick does not accurately reflect what the actual temperature of your body is. So that's an interesting thing to recognize is when you're sweating, it means your fever is breaking. It means you're coming back down.